Hello, I'm Amanda Thomas, and you are listening to A Scientist Walks Into a Bar. This podcast has recordings of talks given at Science on Tap, a series of events held in Portland, Oregon, and Vancouver, Washington. In this episode, we get to hear from Keith Duncan. He's a research scientist in Dr. Chris Topp's lab at the Donald Danforth Plant Science Center in St. Louis, Missouri. Keith was in Portland for a conference just a few months ago in August 2019, and we got him on stage to talk about using X-rays to study plants. He and his team are one of the only labs in the world using X-rays to study living plants. That means they can see plant structures both above and below ground, and we've never been able to see that before. Keith showed a lot of images and animations during his talk, but he gives pretty good descriptions so you don't need to see them to understand what he's talking about. However, check out the episode notes to find links to images, animations, and the resources that Keith mentions at the end of the talk. Take a listen and then find us on Twitter or Facebook at SciBar Podcast and let us know what you think. With that, here we go.
Thanks, Amber. Thanks, Amber, for the opportunity to come out and talk to you tonight. This is an absolute treat for me because for many, many decades, scientists have been dreadful at communication. And that has led to a lot of problems in terms of understanding what we already know and using that as a defense against pseudoscience and wild conspiracy theories and every other thing like that. So I will try to make this as entertaining as possible. I want to thank Scott and Kathleen for putting up with me for a week, but also introducing this scene to me. But Scotty has sworn to tase me if I go too long or if I get boring. So he's not only responsible for me being here, but he's also responsible for making sure I'm good at it. So let's get right into it. X-ray imaging in plant biology, that's a really new kind of thing. So what I want to start with is why plant biology, and specifically where I am, what I'm doing. The Danforth Plant Science Center is the largest nonprofit independent plant science institute in the world. We get to do research on crops that are not commercially exciting, but are called food security crops. They are absolutely essential for many people around the world. But it doesn't make economic sense for companies to do it. So we do the hard work of grant writing, getting funding, getting donations, and we do the work that's crucial. It's in St. Louis. St. Louis is really sort of the center of plant science for the country, in kind of the same way that San Jose is the center for a lot of the .com startups, and Boston for some of the medical devices. St. Louis is where you do plant science. Specifically, I work in the Topp lab. That's Chris Topp, that's actually his name. And we do a lot of root research, and it was the whole idea of looking at roots in the soil without having to rip them up out of the ground, which got us interested in X-ray imaging. What I'm going to talk about tonight, there's 5 other scientists that I work with, and you've heard these speeches before. It wouldn't be possible without the entire crew, but these 5 people particularly played a major role in the science I'm going to tell you about tonight. Why plant science? It's not exciting. You don't have a 5K for weed control. You have 5K for breast cancer, which you should. Dramatic. But why plant biology? It's only important if you want to eat, drink, wear clothes, or have medicine. Other than that, it's not, and breathe. Breathing's pretty cool too. But people are typically plant blind. They don't know the connection between a farmer, when they think of Farmer in the Dell and being 5 years old and singing songs, to the fact that insulin is something that is an ag product. Oxygen that we breathe is crucially produced by plants. Soil is held in place by plants. The CO2 is slurped out of the air by plants, and it's crucial to have. So being in a root lab, we want to use X-ray imaging. So I'm going to give you some background on what X-ray imaging is about. I only slipped once. I'll speak slowly. This is my grandfather's watch. When I joined the lab 3 years ago, I had to figure out how these X-ray instruments work. My background was in microscopy of plant pathogens. Amber and I didn't have a chance to go over that. But I had to learn how to use an X-ray instrument because it was really a new application of technology that's been around for many, many decades. But I had to figure out how to apply that to plant science. So I literally, anything I could grab and fit in the instrument, I was scanning. So some of the history, Wilhelm Röntgen, a German scientist, was the first one to actually manipulate X-rays in a practical way. And that first image is his wife's hand. Hopefully she volunteered, but you can see the ring on her finger. And this was in 1895. Since then, X-ray technology has been used in many, many different ways, some not very useful. In the 50s, I think they had X-ray boxes where you could stick your foot in and see how well your shoe fit. Not a really good, healthy use of X-rays. But some of the history, Sir Godfrey Hounsfield discovered the first, or designed the first X-ray CT instrument, and people would call it a CAT scan. Actually stands for computed axial tomography. And that's where CAT came from. But these days, you're only using computers for everything. So essentially, it's X-ray tomography. That really covers everything from low res to high res. And Hounsfield was given a Nobel Prize for that. The basic offshoot of that is you can use non-destructive testing, non-destructive imaging to see inside people. Much more effective than literally cutting them open, and often much safer, even though you're getting dosed with low-level X-rays. Taking that to plant science, there is an institute at the University of Nottingham in England. And in fact, we were lucky enough to visit there last month and meet with them. They were the first ones to use X-ray imaging in a plant facility. And they were the ones able to generate funding from numerous sources throughout the UK and Europe. Although it'll be interesting to see how that works now with Brexit. It's actually not a joke. That's a very serious concern for the impact it'll have on research, because the European Union has been so collaborative, and now jackass politicians are jumping in the way and preventing this normal interaction of ideas, and it's going to be very challenging. Sorry, soapbox suddenly appeared and I stood on it. But yeah, the Hounsfield facility was the first one to do this. We really modeled ourselves after what they set up. So Chris Topp, the principal investigator in our lab, was able to secure funding from numerous sources to get a large-scale, high-resolution X-ray imaging source. And it started operation, notice the date, precisely 3 years ago today. I was hired and started at the Danforth Center exactly 3 years ago today. It's very, very exciting. And there's a bit of a story behind how I got there. It's interesting. If we have time, we'll go into that. But in general, the way this sort of system works is you'll have a sample in the middle, a detector on one side, an X-ray source on the other. And a bit like shining a flashlight on your hand, the shadow on the wall is the image that we're interested in. So X-rays are projected through the sample, hits the detector. So yeah, this is a 2D digital radiograph, and that's what you get by one quick image. What we'll do is rotate that sample over 360 degrees and collect thousands of these images. And then, and this is something you'll hear a lot, our computer scientists will assemble that into a 3D volume. Now, this is a digital pepper now, so we can digitally dissect this without ever physically touching it. We now have access to information on the inside that you can't get in any other way. If you hack it open with a knife, you've literally changed everything you're trying to look at and measure. So, in addition to my grandfather's pocket watch, I literally went to the market and bought a bag of produce and just started scanning stuff to see how it worked and try to develop the conditions, because there was no instruction sheet. We bought this industrial scale X-ray scanner from a company that sells them to Boeing and Toyota and Apple and Microsoft, where they scan aircraft parts and piston rods and engine blocks. They had no idea what we were putting in there, so they couldn't offer me any advice on what to do. So I put everything in there. So you can look at ears of corn while they're still on the plant. And this is where it starts to get exciting. If you didn't already know, these are obviously ears of corn, but the silks, their role is to capture the pollen from the corn. One pollen grain lands on one silk, the pollen germinates, goes all the way down the silk and forms one kernel. So the more silks, the more pollen, the more kernels. We can look at this developing ear inside the plant long before it ever emerges. The energy levels we're using, and that's always a question, doesn't the X-ray hurt the plant? Not at the energy levels we're using. They're really, really low. One of the first things I did when I got a new job and actually had healthcare again, went to the dentist. well, I'm getting x-rays and I'm now such a geek, I look, oh, what's the energy level they're using? It was more than 10 times what I was using to look at plants. So I felt pretty good that we're not damaging plants. We're also literally having partner plants that don't get scanned. We're always comparing, is one worse than the other? And in fact, it's actually taking them out of the greenhouse that they don't like. The x-ray's just fine. Ears of corn, that's where the pollen lands. So let's look at the pollen generating structure, the tassel. Again, you've seen these in fields all the time. They shed thousands and thousands of pollen grains. They rain down. They're actually very heavy corn pollen. It drops very nicely and doesn't blow around very much, conveniently. But again, that's what we want. We want a great big rain of pollen down on the silks to get as many seeds to grow and develop on every ear. That's called ear fill. Grain fill. How many kernels can we get on the ear? We can look at the tassel long before it develops. And in fact, corn grows like this will be old-fashioned, like an old radio antenna that you pull out and it telescopes out. Here you can see 1, 2, 3 ears that are going to develop once that plant extends. So all of this information we can now get out of plants non-destructively. So when you get a mature ear of corn, good seed fill here. This is field corn or seed corn. This is where you get your animal feed or your corn starch. But again, we now have access to not just imaging, but measurement, and I'll get into that shortly. We also have something that increases the resolution of what we want to look at. That large instrument uses basic, what they call geometric magnification, geomag. Again, flashlight, hand, wall. You can adjust the size just by moving your hand, moving the flashlight, and that's how you get things bigger or smaller, but that's it. The Xray Microscope adds a really important component. We've had it for just over 1 year. There are about 400 of these in the world, and this is the only one located at a plant science institute doing nothing but scanning plants 24/7. We really are in a unique position, which is exciting, but also brings a lot of responsibility. There's a lot that we should be doing because we can. So the Xray Microscope, it is in fact a microscope. You have lenses here. Here's your sample in the middle. This is your X-ray source. So if you recall from the previous diagram, you project your X-rays through your sample, but instead of just projecting it onto a detector, what's happening is your image goes onto the front of a lens. When it hits that lens, it's got a special coating that converts X-rays into light. So now you have light and a regular old lens. That's what gets magnified. So X-rays, light, magnified by your lens, image. And what we're able to do, this is a little soybean floret that will self-pollinate, and lots of soybean pods come out of there. It's only a couple millimeters long, so a dime would cover that. If you put a dime over it, you wouldn't see it. That's the size we're talking about. But with the Xray Microscope, we can generate a 3D volume that gives us extraordinary detail of what's going on inside. So this is just digitally sectioned, but if you want to think about breeding, and that's what I'm going to be talking about through most of the rest of the presentation, we want to be able to improve crops. Amber had some great stats about, we're going to need twice as much food in 2050 to 2070. Well, earlier than that, we're going to have 10 billion humans on the planet. We are losing arable land. We don't have as much. The amount of land used for agriculture is being reduced. We need to get plants to do more. So plant breeding is going to be tremendously important to use every tool at our disposal. If you're going to breed plants, you need to be able to work with traits. If you wanted to get more and better broccoli, that's going to mean maybe more florets, bigger florets. I mean, what does better broccoli mean? And how do you measure that? So a lot of what I'm going to talk about is working with plant traits, measurable traits. Successful agriculture means the improvement of plant traits to suit the local environment. Traits are anything that is improving the plant. So disease resistance, drought tolerance, water and fertilizer use efficiency, yield improvement, and increasing the nutritional value of whatever it is you're growing. These are all traits that have to be measured and improved. And you can't know if you're doing better if you can't measure it. So yeah, measurable is a crucial part. So X-ray imaging is primarily about how do we measure complicated structures. So if I handed you a head of broccoli and said, measure that, well, you might weigh it. You might, put it in a bowl of water with the edges marked and see how much it displaces, maybe get volume, but how would you count the number of florets? How would you know if you're improving broccoli? So we use X-ray imaging for this, and sorghum is a fantastic example. Tremendous diversity in grain sorghum. And I'll tell you a little bit about sorghum just in case that's a word you've heard but can't link it to anything. It's grown in places where corn doesn't do well. It's really drought and heat tolerant. It produces a fantastic head of grain, and the head is called a panicle. Lots and lots of grains, and in fact, you can use the stalks to make a type of molasses called sorghum syrup. It's long been a staple in the South. Again, it's very, very popular in less affluent situations, less affluent countries, and very arid countries, so it's a very important crop. So we can take a single panicle of barley, of sorghum, scan it in the X-ray system, and now we can take that 3D volume, and our computer scientists will be able to measure that 3D volume in ways that are simply not possible to do by hand. So for example, this X-ray scan here, we're now able to map and count where all the grains are. So the most grains are in the middle, as you see by this graph. It's graphing where the numbers are. The greatest number is right here in the middle, with fewer here and fewer here. You may be able to sort of guess that just by looking, but we can actually count it. We can count the distribution of sizes. We can measure the actual size of the grains. So the darker the red, the bigger the grain. So now we can map where the big ones are, where the small ones are, where everything else is. We now have access to measurements that you simply can't do by hand. Grains can only form if the underlying branching structure is allowing you. Is it big enough? Is it wide enough? Do we have enough branches? If we have more branches, do we get more grains? Or do they start to bump into each other too much? Do the grains fall down? Or do the branches fall down? So we also want to measure not just the grains, but the underlying structure. And again, measurement is a difficult thing, but if you scan it with the X-ray and develop a 3D volume, computer scientists can do really sophisticated measurements. Now, Mao Li is a postdoc in our lab, and my description, she is a math magician. She knows more about mathematics and computer science than any human I've ever heard of, and we're lucky enough to have her work with our plant material. So in order to demonstrate the difficulty in counting all the grains on a sorghum panicle, yeah, she took 2 hours and 20 minutes and really wiped herself out. But it really sort of tells you, we know the numbers, we can now do a lot of sophisticated measurements. So again, measurement is going to be the key. Lots of stuff on this slide, there's lots of things going on, but on the stage right, on this side, we can count and map all the grains, and that's what these graphs are illustrating, where all the grains are. We can actually measure every single grain. We can measure the shape and determine what the shape is. And in fact, we can then computationally remove the grains. Mao doesn't have to spend 2.5 hours pulling all the seeds off. We can then screw up the hand device and go back to grapes. Backward is going forward. That's kind of cool. Yeah, so we can actually measure the underlying branching architecture like I talked about. You can now understand how to get more grains on your sorghum. But why do you want to look at sorghum? In addition to being a really useful crop, you can use it for biofuels, you can use the grains, but there's a local distillery. In fact, St. Louis has a lot of local distilleries. Still 630. Everyone knows that the St. Louis has the Gateway Arch. It's 630 feet tall. Still 630 is very close by, 4 blocks away. Most folks don't know that the arch is 630 feet wide at the base. So sorghum is very important, and we're able to measure it in ways that simply were not possible before. For the wine drinkers, we're also studying grapes, and cool to look at. The grapes are lovely and tasty. They make awesome table grapes for eating, and wine grapes, obviously, for making wine. But much more interesting to us as scientists is the underlying structure, the grape rachis. That's the sort of stems left behind. That's what will dictate how many berries you can have, because berries, once they start to interfere with each other, stop growing. So we want to be able to measure that underlying rachis, that underlying stem. So with some colleagues from the USDA and from Michigan State University, and there's Mao, in the image you can see there's a huge variety of grape sizes, shapes, and varieties. Table grapes, they're all pretty much the same. Wine grapes and wild grapes are really, really diverse. And so what we did was measure with the X-ray system hundreds of these, and then Mao worked her mathematical magic, actually in a mathematical protocol called persistent homology. And you could look this up if you wanted to. It's really quite complicated, but very effective at using branching architecture and measuring it mathematically. And by measuring it, she was able to differentiate dozens of different branch types into which many different grape varieties would fall. So understanding how to improve grape growing and grape breeding, understand the underlying rachis architecture. That's what you want to be measuring when you're trying to improve grape varieties. And in fact, this is a great illustration that she made to show you with this particular architecture, grape berries can only get so big, you can only generate so many, and she was able to mathematically lay that out. It's really sophisticated science, all to get you a good glass of wine. Or grape juice. But there's a lot of grape biology that we can look into. So in the spring, grape vines will actually sprout buds, and those will be the vines that grow and eventually produce your bunches of grapes. Well, we can look at this stick. This is actually just part of the bud that shows up in the springtime. We can look at that with the X-ray, and it's really cool to look inside. But grafting is the other part of how you manage grape production. The rootstocks, and it actually learned by living in St. Louis now for 3 years, most of the rootstocks around the world come from Missouri varieties of grape. They make dreadful grapes, but they make very hardy and disease-resistant rootstocks. So you have hardy disease-resistant rootstocks onto which you graft any number of hundreds of different of good-tasting wine varieties. So we want to know, what does that graft look like? What's it doing? How does that knit up? We know that in humans pretty well, but we don't know what it looks like in plants because we've not had a good way of looking at it. Well, we can now look at that. We can look at this graft junction here and actually map the plumbing. That's like the blood vessels. We can map the plumbing as it re-knits, and some of them don't knit properly, and that will tell us a lot about what's successful or not successful about grafting. So again, we have access to biology that was just really not possible in any other way. So far, everything's been really easy. It's all above ground. We can just plop it in the instrument and have a look. Let's start talking about roots. Corn obviously has been produced for about 9,000 years, and right near the end, we did some neat archaeological stuff. But when you're breeding for corn these days, everyone breeds for the above ground parts because you can see them. It's easy to measure, but the roots are very difficult to measure. So for all the breeding history of corn, the roots have just come along for the ride. So now we're trying to find ways to look at root systems. And before the X-ray system in the top lab, what we would do is go out to our research fields and spend a week or two doing shovelomics. You've heard of genomics? This is shovelomics. We're literally using a shovel and digging up plants, hosing them off with a power washer, and then photographing the root systems. You could get some interesting 2D information because even just doing that, the root systems are really, really different. And that root crown area right at the sort of the junction of above and below ground, that's where all the roots come together. So it's an important part. So measuring that and evaluating that was useful. But in 2D, it's difficult to get a lot out of that. So obviously, if we can do 3D imaging of this complicated structure, there's so much more information we can pull out. And again, if we can measure the traits, we can now breed and improve the traits and find out, do we want roots that go wide? Well, maybe that's good for foraging if the ground is hard. Do we want roots to grow deep? Well, that might be good if you need to go try to find water. So all of these traits we need to understand if we're going to work with them. And in fact, we've been able to publish papers that really specifically describe we can measure root traits using 3D and really differentiate between some of the most important varieties out there. And these are not very exciting slides, but they're very important graphs to show that those little groupings of dots are all the same color. That means all of those we're able to differentiate just by looking at the X-ray scan of the root system. We can measure that crown root and have a difference. Another thing that's cool is we had grown corn plants in large glass jars in a gel. So now we can literally see the roots, but only for about 2 weeks before they start hitting the edges. Well, that architecture that we could measure mirrored the architecture that we measured with the X-ray at the end of the season. So we were able to prove that some of the architecture you see early is carried out through the entire season. So these are important sort of building blocks for us. So some of the early architecture that we could measure was mirrored in the stuff we saw at maturity. So we were able to convince ourselves, look, this is important. The next question is, well, how early can we measure roots and actually see differences? Think back to the slide, well, all the dozens of root crowns that are so wildly different. Roots only do 3 things. They can elongate, they can get longer, they can bend, turn a corner, or they can branch. That's all they can do. But they're making those decisions constantly. So we want to understand what's driving the decision making. Do they bump into something and then turn? Or do they bump into something and branch? Or do they bump into something and stop? We need to be able to understand those things in order to take advantage of the great genetic diversity in root biology. But roots are hard to study. They're hard to look at, they're hard to get a hold of in any kind of natural setting. This is actually my in-laws' property in Macon, Georgia. They had a pretty fierce windstorm, a tree right by the side of the lake, and that's about the only way you're ever going to have a look at roots. You just sort of pick up the carpet and have a look. So let's start talking about some of the other food security crops. Cassava is something that is simply not grown in the United States, but it is a massively important food safety crop in a lot of sub-Saharan Africa, also a lot of Indonesia, some India as well, South America. It's a very, very important food security crop. And by food security, I mean, sure, they want to grow corn, they want to grow wheat, rice, and everything else. But when those crops fail because they have extreme weather, and we're getting a lot more of that, cassava is what they fall back on because it just grows in the ground. It's this great big huge storage root, often 2, 3, 4 feet long, and it is for subsistence. What the Danforth is doing, and in fact it was a donation from the Gates Foundation to the tune of $6 million, working with cassava to try to increase the nutrient content. There's cassava that produces iron and zinc and a few other crucial nutrients, but in levels that are too low. Can we increase the amount of nutrients in the subsistence food and make it more valuable? But it's a root crop. How do you look at it? So it's fantastic for pulling carbon out of the CO2 out of the air, storing carbon in the ground. It's a very important crop. So what we're going to do, or what we have been doing, is trying to look at what processes drive the formation of the storage root, because people haven't been able to look at that without yanking the plant up and then going, okay, well, we can't look at that one ever again. And then you yank that one up. Okay, well, that's a little further, but it doesn't relate to what that one did. We want to look at the same plant over time. And we want to do that with a lot of plants. So we understand which are the roots that will get that mysterious signal, which we don't know yet, turns a regular root into a storage root. So understanding that, we can then breed far more successfully. So cassava in the X-ray, here's your digital radiograph, growing in a potting mix. Now, you might think, what does a root look like in potting mix? Well, essentially, potting mix is like ground up roots. So you're trying to look at roots in roots, and it's really challenging to do. Our eyes are good at picking out some of these big swollen things. No, it was kind of there for a second. Hard to pick out. Our eyes can do that, but it's all the same grayscale. So how do you get a computer to do that? Again, our computer scientists are crucial at looking at these volumes and identifying what's root and eliminating everything else that's not. So if you have brothers, sisters, nieces, nephews, children who want to have a phenomenal career, have them go into computer science. There are massive numbers of jobs in computer science. We can now use computer scientists to pull away most of the potting mix, leaving behind these great big storage roots, and then progressively, systematically pull away more of the stuff we're not interested in measuring, leaving behind just these great big storage roots. So we can start to pinpoint when and what roots will be actually doing things. Sustainability, people talk about that without linking too much specific to it. What I want to talk about is perennial crops, the future of agriculture, and what we might be able to do. So the Midwest has had some terrible flooding over the past few months. Farmers have not been able to plant until very late. Tariffs on soybean and corn has not helped. But having massive flooding and traditional agriculture, very often what's happening is all your extraordinarily valuable topsoil is literally getting washed away. So how can we find ways to lock that soil in place. I've mentioned it a few times with the cassava and with the sorghum. We need to be able to lock the soil in place. We need to keep it there. So can we rethink some of our agricultural systems? We don't need to throw everything else out and start from scratch, but what can we do to modify and change what we're doing? So perenniality is the ability of a plant to return every single year without having to reseed. All of our major economic crops are annuals. You have to plant new seed every single year. Important to maintain a good rhizosphere and microbiome down there because it is probably one of the most complicated environments on the planet because of the sheer number of microbes and living things that are down there. You can have very complicated structures, very complicated environments in many places, but none have the sheer number of microbes that soil does. And the more rich soil, the more the microbes. It's an incredibly complicated environment. How can we take care of that? How can we keep that in place? So if we can start to generate crops that produce useful grains perennially, the root systems are much, much longer. They lock soil in place. They really take care of the microbiome. They slurp tons of CO2 out of the air and store it as roots in the ground. It's a crucially important strategy. So we need to find ways to incorporate and integrate perenniality. So these are just some useful infographics. Allison Miller is a research scientist at the Danforth. She does a lot of work in perenniality. At the end of the talk, there's going to be some websites that you can take pictures of and go there and learn a lot about what's going on. But rotating perennial crops with standard crops, it helps to reinvigorate the soil. It helps keep the soil microbiome vibrant. It helps with erosion. If you have a much more substantial root system, the soil isn't typically getting washed away. And in fact, if less topsoil gets washed into the water, you have fewer algae blooms. But yeah, you get much cleaner water if you can let the plants do the filtering and keep all that in place. And again, it just improves the entire microbiome. So if you just in stages, if you start with some perennial plants who improve the soil, you can encourage symbiotic relationships between microbes and roots, and I'll talk about that a little bit more, because we are getting into that. And it's a really effective way to just improve, in general, what's going on. Is it scalable? You're not going to feed the entire world just doing this, but this is a great tool to add to the arsenal that is going to be required to feed 9 billion, 10 billion people in the next 20 years. Kernza is the commercial name of a perennial grass that produces a very useful grain. It also produces a wonderfully deep root system that stays around for a while. And since this is Portland, this is Patagonia Provisions has worked with Kernza suppliers from the Land Institute there in Salina, Kansas. And Hopworks Urban Brewery right here in Portland, I think there's one in Vancouver as well, they have made 2 different beers featuring this Kernza grain. It's not a gimmick, honestly. It's actually something that it's a very good beer. I had to make sure I tried a couple. No, it's a fantastic beer, but it's also a great idea. And we need to get a lot more breweries doing that. And in fact, Allison and I met with some of the local brewers in St. Louis to get them interested in using Kernza and other sorts of perennial grains, not as the primary ingredient, because that's not how you make beer. But if you can get them to enhance the flavor or change the flavor or make it interesting, that's definitely something we want to pursue. So we go to a lot of breweries for our job. And test a lot of things. It's a great world where you can have good beer in cans, good wine with screw caps, and small distilleries all over town. We live in a good world, more often than not. So what we're doing in our lab is we're looking at switchgrass. This is a perennial grass. It doesn't necessarily make a grain, but it is a fantastic way to sequester carbon, pull CO2 out of the air, because we really, really need to do a lot more of that, and store it in the ground. Not only does it secure the CO2, but it enhances the carbon value of the ground. And so we're working with folks from the University of Texas, University of Florida, with some grant money from the USDA and the Department of Energy, because they're very interested in bioenergy sources. And we're now able to scan this entire 1 meter tall segment. That's not even the whole root system, it's just part of it. But we're now able to measure this, although when I gave this data set to our computer scientists, they said no way, because this one data set alone was about 700 gigabytes. just of raw data. And you need serious computing power to juggle all that. The other end of the size scale, we're very interested in alfalfa, but it's a root system that we are able to segment, and that's actually the computer term for separating what you want to see from the rest, because X-rays themselves are dumb. They go through everything. They're not going to tell you what's the pot and what's the air, what's the soil, what's the water, and what's the root. They just show you everything. Our computer scientists have to pull the biology out the other end, and that's why I keep talking about them over and over again. I just do the scanning. I get the pretty pictures. They do the work. They pull the information out of the scans that I generate. Alfalfa is a really important crop. It's used a lot for grazing animals, for forage. But what it is very good at doing is it has established a mutualistic relationship with fungi that live in the soil. These fungi will grow into the roots, colonize the roots. The fungal hyphae, the fungal mycelium, will grow way out into the soil, far beyond the roots, pull in phosphorus, pull in water, give that to the plant in exchange for the sugars that the plant makes in photosynthesis. So those sorts of relationships are really important. So we're able to study that root structure and look at, is the root system better or worse? Is it different when it has been colonized or not colonized? So these are the questions that we're looking at. Other interesting microbes, there are rhizobacteria, root-dwelling bacteria that can fix atmospheric nitrogen. So I think it was around the early 1900s when the Haber-Bosch process was developed to basically break up atmospheric nitrogen and generate fertilizer. When you can generate chemical fertilizer, you can now grow many, many more plants, better plants in many different places. It is a tremendously inefficient process. But plants can't use atmospheric nitrogen. I mean, we're breathing it all the time. It's about 70% of the atmosphere. We can't do anything with it. These bacteria have been around for millions and millions of years. They pull nitrogen literally out of thin air, convert it to forms the plant can use, and hand it off to the plant, again, in exchange for sugars. Sugars are made by photosynthesis. So that's a mutualistic relationship that's been going on for as long as there have been bacteria and plants. And in soybean here, we used a large format X-ray system to scan that, segment out the root system, and in fact, identify where the nodules are, and then use the Xray microscope to actually look at one individual nodule. So now we can start to study this interaction literally on the cellular level. So the more we can understand about these mutualistic relationships, the better chance we have of getting plants to sort of fend for themselves. Rather than dumping on tons of chemical fertilizer, can we get the nodules to help the plants do this themselves? Other examples, again, I talked about mycorrhizal fungi. These are the fungi that, again, colonize roots. They do it to corn, they do it to alfalfa, many different plants. And what they do is they extend the range, the reach of the plants. So if we can study this interaction at the cellular level, we can see what's going on inside. We can have a better idea of what's a good relationship? What's a bad relationship? When does it start? Are there cheaters? Are there ones who pull out sugar but then don't deliver? How does the plant deal with that? There's huge game theory ideas on what goes on. The cheaters are tossed out really rapidly. And the altruistic ones die rapidly, so you can't just keep giving, you'll die. So it's really a fine-tuned interaction, and we're trying to understand what's driving that. So we're able to use the Xray microscope to look at, here's one root cell that's been colonized, and we can work with that 3D volume. So it's possible now to look inside maize roots and evaluate and measure this interaction. Now, again, measurement. How would you measure this? What are you supposed to look for? When I don't get teased by Mao and by Ni, who are our computer scientists, about, that scan's terrible, we can't see anything with that, so I'll go back downstairs and go back down and do another scan. But when I give them good data, especially something like this, they can computationally isolate the fungus, which is seen here in yellow. You can now count those, you can measure those, you can evaluate the extent to which the roots have been colonized by this beneficial fungus. So again, it's a bit of a broken record, but that's what I want you to leave with. We're now able to measure things that we could never even think about measuring before. I'll just sort of mention briefly one by one some of the other interesting types of plants that we want to study now that we can study things in very interesting ways. I'd never heard of this until 2 weeks ago. So that's how new this data is. I literally got it before I left. Teff. I spelled it wrong when I first got it. But it's an ancient grass from sort of the Horn of Africa, the Eastern Africa, Ethiopia, and Eritrea. It's very hardy. It produces a huge amount of grain. It's a very, very small grain. And the grass is also used for forage. But again, any sort of grass that grows very well in hot, arid climates, we want to know, how does it do that? And can we take advantage of those traits and move them into more commercial varieties? Or can we move that into perennial grains? So anything we can learn about these plants. And the structure we're looking at here, this whole thing was barely 2 millimeters tall. So if you stack 2 dimes, you've gone over 2 millimeters. So it's a really, really tiny plant. Interesting source of material. Duckweed, I know you've seen duckweed. If you've seen ponds, if you've seen puddles, if you've seen the ditch on the side of the road with water, you've seen duckweed. There's dozens of varieties. And this one is one of the ones we're working with now. It's a really good source of protein. You can grow it in lots of large kind of fermentation vats. Great source of protein. It's also really good at toxic remediation. You can sort of throw this into sewage runoff or chemical runoff or slag pits, any sort of thing like that. And very often the plant will simply develop the mechanism to pull in and isolate the toxic compounds. Ceteria is another sort of, it's the lab rat. One of the lab rats in plant science. Arabidopsis is the other one, but it's so unrelated to anything else. This is actually a grass. It has a lot of genes similar to corn and sorghum. So if we can understand a lot of the sophisticated structural details of foxtail millet or Ceteria, we can take that knowledge and apply it to corn and sorghum and maybe extend it to wheat and rice and other grasses. Again, this is Ni Jiang. She is our other math magician. She took the 3D volume that I did, identified the entire structure, and now has computationally identified and counted and measured every single developing structure in there. That's exactly the power that we need computationally to start to bring meaning to the scans. Again, I said in the beginning, it's all about measurement. We don't know if we're making it better if we can't measure it. Some things are easy to measure, height, width, numbers. But a lot of things, especially early on, are very, very difficult to measure. And we want to measure early on. The earlier, the better. This is actually just 15 days after the seed germinated. It's already this complicated. The earlier, the better. We want to be able to measure things. Booze. Lots of other cool things to measure. I went to one of our local breweries to see if I could get some hops buds for scanning, and in fact, they said, no, dude, we use pellets. Well, they're not very interesting, but they did actually go to some home brewers who had hops growing in the backyard. And what I thought was so interesting, having never looked inside one before, there's not much structure there. This is the only solid stuff. The rest are just overlapping leaves. So a lot of, and they're incredibly fragrant. If you ever had hops, it's just an amazing, amazing scent. But all of that comes from glands that are producing oils in the central area, and then it just sort of schmooze out on the leaves, or the bracts or the sepals or petals, whatever you want to call them. And that's where the sort of scent comes from, and that's why they're so useful in brewing beer. Never would have known that without grabbing stuff, throwing it in the X-ray system. This one was cool. Yes, it's just a corn kernel, but the detail we can look at is remarkable. And these kernels are from Stumpy's distillery in Illinois, right across the river from St. Louis. This is a distillery and farm, and they produce fantastic whiskeys and it's not just locally sourced. I mean, it is literally only from their property. So we had a lovely tour there, and these are kernels that I borrowed and scanned. Whiskey barrel stave. Nearly every single barrel that you'll have for whiskey, for rye, for all sorts, has to be Missouri white oak. And I was very thrilled to actually go to Missouri to realize I'm now going to be just about the center of the whiskey world. And in fact, the Missouri Whiskey Society, it was formed at Gamlin Whiskey House. I'm now an ambassador for the Missouri Whiskey Society, which has some tremendous perks. But sort of in exchange, Yeah, this is just a habit. I mean, it's just something I enjoy doing. I can stop anytime. But they gave me a barrel stave. In fact, one of their events, they had a barrel stave painting event, and I'm hopeless at painting, but they gave me this barrel stave. And this charred layer is what's on the inside. And when they char barrels, what that does is sort of helps the plant material, all the boards in there, to release some of the tannins, some of the other esters and polyphenols and other exciting names. But as the whiskey penetrates, it gets in and out. As the temperature gets hot and cold, it penetrates, comes out. You lose the angel's share to the barrel. But it's a cool process that no one ever gets to see. So I thought it would be kind of fun to take this barrel stave, slice it down a bit, and have a look at it with the two different instruments. So this is actually the surface that is charred. And at this resolution, it's not that exciting, but what you can see is the thousands and thousands of holes. It's just this massive plumbing system with just hundreds of miles of very, very tiny conduits. And a lot of the liquid can travel in and out of there, pulling flavor out of the wood. So that's relatively low mag. Of course, I say, ooh, let's up the mag. So again, this is the surface that's charred. And this is what's called a fly-through. We're simply frame by frame going through all of the entire volume. And again, you can see it's massively loaded with holes. And then the whiskey will get into all this area and just penetrate in and out, and that's where your flavor ends up developing. So that was kind of cool to think of this wonderful amber nectar of the gods in terms of where it comes from. We'll start wrapping it up, and this is kind of the National Geographic moment. We were really, really fortunate to work with some colleagues from the Archeological Institute in Mexico, who originally in the 1960s discovered some caves in central Mexico where it was really arid, meaning if something was dried 9,000 years ago or 5,000 years ago, it's not going to fossilize, it's just going to sit there. So they found some corn samples that were between 1,500 and 5,000 years old. And we were fortunate enough to patiently wait while Jean-Philippe, not at all a Mexican name. They were Belgian, they moved to Mexico. It's bizarre. But Jean-Philippe was able to work with the Mexican government because these are national treasures. He was able to bring them to St. Louis because of the X-ray imaging we were doing to do non-destructive testing of these phenomenal archaeological samples. So this was massively exciting. So we put the larger, younger sample, it's only 1,500 years old, put that in the large instrument, and it's remarkably similar to that exact same region that we look at today. So a lot of the structure is very, very similar. And this, again, our computer scientists now have this, and are working with this to start finding out what can we measure, what's important to measure, what's meaningless to measure, so that we can compare this 1,500-year-old sample to modern material and see what sorts of things have changed, what's the same. To look at it by eye, that's going to be really difficult to do, but if we have sophisticated numerical mathematical measurements, that'll be fantastic. That's the young one. Let's look at the 5,300-year-old sample and put that in the Xray microscope. And I wish I had the clip here. One of my X-ray technicians, Gus Tees, I was so carefully moving this, and he suddenly played on his phone the theme from Indiana Jones. And I just started laughing. I had to back up and start again. So thank you, Gus. But this one is really interesting. This is the crown region, or the nodal plexus. This is where all the roots come together and all the plumbing comes together and then joins up into the rest of the plant. There's a lot of really interesting bright dots. And when you're looking at a 3D volume, if you see dots over a brightness over a long time, that means it's a long single structure. There are structures called phytoliths in modern plants. They're made up of silicon. Very often they're made to dissuade herbivores from eating. If you have lots of silica and the teeth eat it, they end up grinding the teeth away, so the plant or the animal learns not to feed on that plant. It provides structural strength when there's drought. So if you lose the water turgor pressure and the plant wants to wilt, if you have some stiffening agents like these phytoliths, that's very helpful. So we're actually going to work with some paleobotanists from University of Michigan who have been looking at million-year-old phytoliths and say, look, is any of this familiar to you? Do you see some of this? And compare it to modern maize varieties. What's also cool, and this will sort of wrap up the plant part, Teosinte is a wild form of corn, which is still growing in the central highlands of Mexico. That's the original plant from which our ancestors 9,000 years ago first generated what's now modern corn. You can see modern versions, you can see the old corn, and now with some of these archaeological samples, we can start to look at the progression. How did one go to the other? We know what genes are involved, and we can sequence both. That's actually relatively easy. There's only been about 2 or 3 major gene changes that made modern corn from this incredibly weird-looking bush that made, dozens and dozens of heads. So can we track some of that now with looking physically and directly at these archaeological samples? I think we just have a little bit of time left. I want to make sure we leave time for questions and comments. But it's interesting to know how I got to where I am now, because a lot of folks just don't know sort of the path of science. Scientists are always white men with beards, and they sort of walk around like this, and they do wacky stuff. So in 1990, I finished graduate school at the University of Georgia and was hired as a research scientist to use some of the most advanced microscopes on the planet to study plant pathology, plant diseases. And it was just, I grew up in Detroit. We had thousands of elm trees. They all died because of a fungal disease, and I was hooked. I didn't even realize plants could get sick. So I was so stoked on using microscopes to study plant diseases. Lots of fun. I was able to work with, again, some of the most advanced electron, laser, fluorescence imaging equipment to study plant pathogens, things that cause disease. So these are just very cool black and white images on film. I had to learn how to use a darkroom before any of my science would actually happen, so that was a lot of fun. But these are pathogens on leaves. And if you've had any gardens, if you've ever seen that sort of fuzzy white stuff on the top, that's powdery mildew. Lots and lots of different ones. They infect most leaves. So powdery mildew on wheat and barley, powdery mildew on grape. This is a rust fungus that attacks soybean. It's a huge, huge problem in Brazil, where just about most of the soybeans in the world grow. And in fact, what's really cool is with this type of electron microscopy, it uses liquid nitrogen. You can actually see that this plant cell has collapsed. That's the one that the fungus invaded. So the spore landed there, germinated, and put all of its guts into that, hit the cell, and that cell collapsed. And in fact, the fungus uses a glue to make sure it can stick to the surface. Now, these are wax crystals. This is why water beads up on leaves, because it's just loaded with these crystals of wax. So when you have a lovely, droplet of water, it's because it sits on those. That's a great way to keep fungi from sticking to your leaf, unless it can overcome that by making a glue. Cool, cool stuff. The fungus grows through the plant, and like alien, it just rips its way out through the other side and disperses spores, and it's really cool. So I had an awesome 25-year career, even though it was in Delaware. Was it Wayne's World? Hey, I'm in Delaware. Fantastic career, though very exciting. More than half my life was spent doing that. Turned 50 years old, and then suddenly, bang, gone. Disappears in a puff of corporate mergers. I got laid off. For Christmas, of course. 2015. Merry Christmas. Merry effing Christmas. So we had to leave Delaware. There was nothing left. So we looked around the country, and I kind of started off by saying, the Danforth Center is in St. Louis. We knew the Danforth Center existed. It was the place for plant science. The Salk Institute and the Howard Hughes Institutes, and the Mayo Clinic. Those are the best places in the world for human medicine. Plant science, St. Louis. So we rolled the dice. We sold the house. We just packed up and moved to St. Louis. No jobs, no interviews, nothing. We moved. And I figured, all right, I have a severance package. I can find something to do in the 6 months that I had. And as it turned out, I was hired within days of getting there just because of good fortune. We were in the right place at the right time. Yeah, so that's a bit of the background. It's precisely 3 years ago today I started working at Danforth, and to be honest, I'm thrilled, I'm honored, I'm humbled. It's the kind of thing that you realize you're in a unique position, and you can't modify the word unique. No one else on the planet has 24/7 access to an X-ray microscope to do nothing but study plant biology. So it's daunting, it's exciting, it's a lot of fun. And I thank you all so much for coming and for your attention.
Raise your hand, I'll walk around. Ooh, this is gonna be daunting. Let's see how many I can get to.
You showed that you can take trees and vines and they can do grafting, and then you showed the roots of some of the perennial plants, and especially the big ones. Mm-hmm. Is it possible to take perennial plant roots that are really hardy and drought resistant and then graft the agricultural plants onto those root systems?
That's a great question. If you didn't hear the question, it was, can we take the perennial root system, which is the great advantage, can we sort of graft on the economically important part on the top? Right now, no, but because they're all grasses, they're very closely related. So we want to understand what causes perenniality, and can we take those factors and actually move them into root systems that are around for a long time? Great question.
All right, we're right here in the center.
Awesome work. My question was a lot of it, like with the branching problems. Mm-hmm. Are there any optimization that you're looking at? Right. So you'd say the grapes would interfere with one another. So is there some optimization of the branching that you could do?
Yeah, that's a great question. What are we doing to optimize branching structure relative to berry formation? Step one is to map the branching, which we did. And now what Mao has done, in fact, again, over the past few weeks, we have scanned, we just, again, went to the store, bought loads of different types of grapes and scanned them with the berries. And she has developed a mathematical model that looks at what they call steric hindrance, or the stereo-based interference of when things bump into each other. So she's now using one mathematical model for the rachis, another model for the berries, combining those, and this is math that's just way beyond me. But what I do understand is plant pathology. The more that grapes touch, the more disease can develop there, because you get a little microclimate that's great for mildews and some other things that are terrible. So that's how Mao and other scientists are dealing with this, combining those two factors to sort of map out that structure. Good question.
All right, we've jumped the aisle. We're right here.
So in recent times, there's been a big increase in non-invasive medical technology. So I was wondering if it was the same for plants. Do you know if any labs are using like sound or vibrations of roots and soil to help with plant biology and not just like ultrasound?
We're doing a lot of different kinds of non-destructive imaging to look at plants. So, like I said, X-ray CT for humans has been around since the 70s, and plant biologists have wanted to do that since the 70s. But that means, okay, take your plant, throw it in the trunk of the car, tip it over, drive to the med school, hope you can bribe someone to give you time, tip the plant, dreadful. So that's why only very recently have plant scientists had more regular access to X-ray CT. MRI is really good for following liquid flow, as well as, and this is a bit more kind of out there, neutron imaging. There are various labs around the world that do neutron imaging, which allows you, again, to do follow liquids as they go through. Ultrasound is a really cool one that you mentioned. One of the projects we're involved with is trying to get out to the field to actually look at roots in a field, because right now no one on the planet or any other planet can do that. No one can look at roots in a field and actually see what's going on. So what we want to try to do is, can we do ground penetrating radar and actually bounce it off the roots and see something? Can we use backscatter X-ray systems, again, to sort of bounce X-rays off these things? On the one hand, yes, we can. On the other hand, the best you can do is like find a lost rake. So the resolution's really crappy. But that's where the research is going, and that's also part of how we get our funding. We get grants from what's called, you've probably heard of DARPA, which is the Defense Advanced Research, when they just fund this wild-ass stuff just to see what might work. Well, ARPA is the same thing for agriculture. So we're getting some interesting funding to try to see how can we push development. And so we're working with detector manufacturers, X-ray manufacturers. How can we get that technology into the field? Because that's ultimately what we want. Everything we're doing is well, okay, the field's over here and the lab is over here. We want to get as close as possible to the actual field. So yeah, lots of different things that we're looking into.
Portland is full of liberals who want to do what's right for the environment.
Yeah, and there's one more here.
But those same people are like, we're going to buy non-GMO food.
Right.
So what do you say to those people?
What I say to the people is, look who's trying to sell you something. So if someone is saying, we've got non-GMO grapes, and we want you to pay 75% extra for that. Okay, what's my alternative? What about the non-GMO? Or what about the GMO? Oh, there aren't any other varieties. So that label, that certified non-GMO label, actually doesn't provide information. It's crucially important to want to know what's in the ingredient list. You absolutely should. That label doesn't tell you anything about it. So a GMO is not an ingredient, so you don't have GMOs in your food. It is a technology used to produce the food, and the final product is chemically, down to the atomic level, indistinguishable from food produced with genetic engineering to food produced without it. That's a really long explanation, and that just doesn't fit on a sticker. And it's really complicated. And that's part of why science has done such a lousy job of communicating. You could go to every scientific society in the world. These are the reputable professional groups that are fanatically self-policing. And every single one is unanimous in the confirmation that genetic engineering produces food every bit as safe and allergy-free as conventional. And that's the science behind it. And you know, the same people who might say climate change is real, we are absolutely heating up, which we are, follow the science. That same person will then go, but GMOs are dangerous. Well, no, they're not dangerous. They're no more dangerous, no less dangerous than what's already out there. They don't cause more allergies. They don't cause less allergies. It's exactly the same product in the end. The technology is how the plant's generated. People would say, we're killing butterflies because we have this toxin in plants, and butterflies land on the plant and they get killed. Well, no, butterflies don't eat roots. The Bt toxin is produced in roots, and only root insects and only actually a small family feed on those roots. So there's a lot of drama, there's a lot of emotion, and it is terribly important. So again, I urge you to go to some of the websites. I don't know if you can put it up on your website, that list of websites I had at the end of the talk.
We could probably do a Facebook post.
Okay, perfect. So yeah, look for the Science on Tap and look for the Genetic Literacy Project. They really combine a lot of the stuff in a very objective, informative way. And again, it allows you to ask the right questions. Don't go for the vague and ambiguous. Go for some details, and ask the questions.
All right, we got time for two more. One of them's right here.
Thank you. I wrote mine because I'm a little nervous speaking. So botany and agriculture science are unfamiliar topics to me, so I'm sorry if I don't use the right terms. But you mentioned earlier the idea of changing farmland to mimic nature a little more.
Mm-hmm. Yeah.
To that end, are you scanning plants that grow together to see the structural relationship at the root level?
Wow, that's a great question. Yes. Even if you just think about, you probably would never have thought about it before. 100 years ago, when we would plant a field of corn, the corn was like at least a yard between every plant, and you just didn't get many plants per acre. What you look at now, you can barely put your foot between corn plants because they're so tightly packed. And so the root systems are interacting like crazy, and they're all exactly the same. We know nothing about that. So we are doing work just within a cornfield to look at that, but for the perenniality and for sort of bringing things back to a much more sustainable, non-monoculture environment, there's a huge amount of interaction that we want to study. So yes, that's a great question. So we're looking at the cellular level, looking at the microbes interacting with the roots. We're looking at the root level, and this drives Mao and me absolutely bonkers because it's so difficult to just look at one root system. Well, if you have roots intersecting and interacting, really complicated. But that's a great question, and that is really a big part of what we want to get into sort of now and in the future. How do plants, especially the diverse plants, interact in a field, in large scale, small scale, and anything else?
Let's give it up for Keith Duncan.
Thanks for listening. We're on Facebook and Twitter at SciBar Podcast. That's SCIBAR Podcast. And we'd be super grateful if you would consider posting or tweeting about us. If you're into that kind of thing, reviewing us on Apple Podcasts would help too. If you're in the Pacific Northwest and want to come to an event, check out our website at scienceontaporwa.org. And that last part stands for Oregon and Washington. You can also find out more about events on Facebook, Twitter, and Instagram at scienceontaporwa. As always, I'd like to say a big thank you to my amazing group of volunteers. Some of them have been helping run events for more than a decade, and this would not happen without them. They are Scott Fry, Chris Gowan, Sam Lauk, Rita Nygren, Steve Perry, and Chris Trone. I'd also like to say a special thanks to Amber Peoples for running things for the past few months, and to Graham Tully for his excellent audio production. Finally, a big thank you to Jonathan Coulton for letting us use his song Mandelbrot Set as our intro and outro music.